I'm going to talk to you about models of pregnancy. And I think the first thing I'd like to start off with, like every other speaker has kind of begun with, is what are the major challenges in perinatal medicine. I don't want you to take these home as the only two challenges we have in perinatal medicine, but these are definitely the big hitters. When we start talking about premature birth, we are talking about 1 in every 10 babies that is born prematurely or before 37 weeks. In addition to that, intrauterine growth restriction in developed countries will end up being about 8%, 8 to 10%, again about one baby per birth. As you get into more developing countries, this can increase up to around 24%. So again, leading to major causes or major challenges in perinatal medicine. The primary cause for intrauterine growth restriction is going to be placental insufficiency. And that's the one I'm going to talk to you about quite extensively today. But I should point out that premature birth, just because we don't know everything about how to, supply nutrients to these babies or to these infants now, they actually don't reach their genetic potential by their, expected birth date. So again, these premature infants may be intrauterine growth restricted, and one of the main effective ways of eliminating, intrauterine growth restriction is to essentially deliver that baby early. So again, they interact or they influence one another quite a bit. There's more to this than just the perinatal morbidity and mortality. There are going to be lifelong consequences. And just to demonstrate that, that really does wiggle up here. So just to demonstrate birth weight is associated with impaired glucose homeostasis in the long term. Again, if you look at birth weight, that baby that's born about 3.5 kilos is going to have a much lower risk for developing diabetes than an infant that, say, is intrauterine growth restricted or somehow has its placental insufficiency so it can't meet its genetic potential. On the other side of this scale, you can look at macrosomia, which occurs quite often in diabetic or obese patients, leading to increased birth weight and again increasing the risk of diabetes. So this leads us to the Barker hypothesis that I know we've pointed to earlier today as one of the risk factors, but what we really want to look at is the adaptations to the cell structure, the metabolism, and physiology, and they're permanently altered during development, either in response to nutrient restriction or in response to overnutrition to lead to these lifelong risk factors. And this doesn't always play into diabetes. You can also have coronary heart disease, hypertension, and several other diseases that may be impacted metabolically long term. So I was, to kind of begin to think about the models that we have for pregnancy. Of course, Dr. Ireland gave a beautiful slide showing, the number of researchers that use small rodents is in that 80% range. So obviously these are very inexpensive animals to use and house. However, they're altricial, meaning that they're born, most of their development happens postnatally, or aspects of their development will happen postnatally. So they may be limited in what we're really looking at unless you include lactation into your growth restriction or understanding the pregnancy. In addition, you can genetically modify, you can do a lot of important transgenerational studies, but you are limited at actually looking at the metabolic flux within the placenta or across the placenta and within the fetus. That leads us to some of these more precocial animals, the guinea pig, the non-human primate, and the sheep in particular, that we can use to study programming events. Now, of course, non-human primates are the most genetically identical to the human. Obviously, that would offer a lot of benefits. However, they're very limited in what you can do just due to the expense and extensive experimental procedures that are required, leading to only a very few number of animals. Also, it's very difficult to maintain a non-human primate and actually put catheters into the fetal vasculature. The guinea pig is, of course, precocial, but is also difficult to catheterize to really study nutrient fluxes. So that leaves us with the sheep and one of my favorite cartoons by Gary Larson. But I should point out, and I'll highlight the attributes to the sheep, but what I wanted to point out here was in addition to being able to understand fetal physiology and placental physiology, which has the sheep has offered research on for the last 40+ years, there are several models of intrauterine growth restriction that span from nutrient deprivation to the mother, or nutrient restriction in the mother, to actually placental insufficiency that's created either through the carunculectomy, removing the placentation sites within the uterus before the animal is actually pregnant, or hyperthermia, which is creates placental insufficiency, and when you expose this mother to high ambient temperatures, essentially a model that was identified in northern Australia. So the real question is, why do we use the sheep? And that's usually what I get asked. And what I want to point out is that the sheep is really the only fetal model for studying normal or regulated in vivo metabolism under normal or regulated, non-stressed, unanesthetized conditions. And these conditions almost require surgical placement of catheters, both in the fetal circulation as well as in the maternal circulation. So with that in mind, I just want to show you what we're talking about, and that we do have the ability to go in and manipulate the fetal uterus without actually causing abortion post-surgical procedures. So here we've now done a hysterotomy. We've exposed the uterus, exposed the fetal hind limb. You can see we've now made a small incision within that hind limb, isolated the saphenous artery. Within that, we've now put in a catheter that we can use to, in this case, sample from. We can use venous catheters for infusions. We then would close up our incision, both on the leg and the uterus, put the uterus back into the U, close up that midline incision in the U, and then tunnel those fetal and maternal catheters to a pouch located on the side of that U. And here you can see a U in one of our metabolic crates. Again, she's dog-collared here to just kind of keep her from spinning around in the crate, but we have food and water up front, and you can see that small mesh pouch on the side where we can gain easy access without really disrupting that U or having to chase her around to catch her in this environment to study a fairly unstressed animal. So with that in mind, what I'm going to turn to is some examples and hopefully try to also look at the storyline that we've been developing over the last 10 to 15 years on how catecholamines are impacting islet development. And so you can consider that the placenta is the conduit between the mother and the fetus that is essentially allowing oxygen and nutrients to transfer and support that growing fetus. However, in cases where the placenta fails and becomes insufficient, the nutrient oxygen transfer is decreased and fetal growth succumbs or is slowed. In response to these lower nutrient and oxygen supply, the fetus has low glucose and low oxygen concentrations, which then stimulate a stress response, increasing their circulating catecholamine concentrations. And the story that we've been working on is trying to understand how these catecholamines can impact skeletal muscle or adipose tissue, those insulin responsive tissues, or actually impact the islets of Langerhans, specifically the beta cells, to understand how insulin secretion is altered in response to not only growth restriction, but also high catecholamine concentrations. The other caveat to this is it's not an acute response like you would expect in an adult, where you see that bear, you flee from the bear, your catecholamine levels go down. In this case, we're looking at a very chronic response where over time these fetuses have sustained hypoglycemia and hypoxia and sustained high catecholamine concentrations. And that will become apparent as I go on with what we've been doing. As I mentioned, the hypothermia model leads to placental insufficiency-induced intrauterine growth restriction. On the left-hand side, I show the growth patterns of both the fetus and the placenta. In the blue are the normal growth patterns for the sheep, and in the red would be the intrauterine growth-restricted fetal growth patterns. What we do is expose these ewes to high ambient temperatures, about 104 degrees Fahrenheit during the day, about about 95 degrees Fahrenheit at night, 35 to 45% humidity is enough to significantly reduce placental growth between 40 and 95 days. Looking at this growth restriction, what we're really trying, or looking at this heat stress impact, we're really trying to restrict placental growth, and that leads to further placental, not only a smaller placenta, but further placental functional impairments later on, where you then begin to see the drastic reduction in fetal growth at later stages out here around 140 or 135 to 140 days. Again, as I was mentioned this morning, the sheep pregnancy is around 145 to 149 days. Again, we have smaller placenta, smaller fetus, abnormal umbilical artery blood flows. The fetus is hypoglycemic and hypoxic, and this is not impacting the maternal milieu. So she still has normal glucose and oxygen levels. They're just unable to get to the fetus, which increases the gradient and actually is probably a sparing mechanism to increase transport. The other thing to consider is the perinatal mortality, which is increased in these fetuses. So now to just kind of put some numbers to those general statements I've made, if you look at fetal weights and placental weights at 135 days or 90% of gestation, they're reduced about 50 to 60%. If you look at the oxygen content, again, oxygen content is reduced around 50%. So they are hypoxic. In this talk and for the rest of this talk, I'll be talking about a glucose-stimulated insulin secretion challenge. And what we're doing is essentially performing a square wave hyperglycemic clamp to evaluate insulin secretion in the sheep fetus that's either a control animal or an IUGR animal. In the unstimulated condition, we have about a 50% reduction in glucose concentrations. We increase those plasma glucose concentrations for the controls to about twice of normal. And then we bring the IUGR animals up to that too. These concentrations are about 90% of maximum stimulation for the sheep fetus. What we're really interested in then is what's happening to insulin secretion. And it should be pretty obvious when you look at the control animals in blue to the IUGR animals that response in the IUGR animals is marginal at best over that square wave hyperglycemic clamp. So the response is significantly blunted. In addition to just having this placental insufficiency model, we've also created models where we've infused insulin into the maternal circulation, lowering the maternal glucose concentrations, which then lower the ability of that mother to supply glucose across the placenta. If we make the ewes chronically hypoglycemic, and then look at them 2 weeks later, we again see a decreased insulin concentration and impaired insulin secretion response to that square wave hyperglycemic clamp. But the reduction is about 45% or half of that of what we saw in the IUGR animals, which brings us to the question of what factors may be influencing insulin secretion. We know glucose and oxygen probably play a role, but the other thing that we've been looking at extensively is norepinephrine or catecholamines. Norepinephrine concentrations are negatively correlated with oxygen content, and if we compare the control animals to those IUGR animals, we see about a 4-fold increase in circulating norepinephrine concentrations. Now this is happening near term. So catecholamines may be impacting beta cell dysfunction, either by suppressing it or causing creating a programming mechanism that permanently alters islet function. I know most of you probably aren't beta cell biologists, so I just wanted to put up a quick slide to remind you that glucose enters the beta cell, that glucose is metabolized, it then increases ATP to ADP ratios. Those as that increases, it will close the potassium channels, depolarize the cell, allowing calcium to enter and insulin to be secreted. In addition to that, this trigger per se through glucose, we also have modifying G protein coupled receptors, and they can be both positive or negative in response to modifying insulin secretion. The one, of course, we're interested in is the adrenergic receptor alpha 2, which has been demonstrated to actually be the one in the sheep fetus that inhibits insulin secretion as well, similar to what you see in the adults, mouse, rat, human, everything else. So I'm fairly simple-minded. I try to do simple experiments. So I put this together as placental insufficiency creates that hypoglycemia and hypoxia and increases catecholamines, norepinephrine and epinephrine, predominantly norepinephrine in the fetus because PNMT doesn't rise until later in gestation when the cortisol concentrations increase. And these are suppressing glucose-stimulated insulin secretion. So the first study that we performed was just to simply use adrenergic antagonists to stop the action of these catecholamines and look at the response to glucose-stimulated insulin secretion. And what we found was increased glucose-stimulated insulin secretion, but this response was actually greater than that in the controls, suggesting that we have a hyper-responsiveness once we've removed the inhibition of catecholamines. This also demonstrated that those chronically high catecholamines aren't being desensitized at the beta cells and that they are still suppressing insulin secretion, leading to decreased growth rates. The other way to do this was to go in and remove the adrenal medulla, which we have shown is predominantly the site where norepinephrine is released in response to hypoxia stimulation. We did this at 98 days of gestation and then came back and evaluated glucose-stimulated insulin secretion later on. So before these fetuses become growth restricted, we remove the adrenal medulla and we look at and we decrease the catecholamines and increase the insulin secretion later on in gestation. Of course, they're still hypoglycemic and hypoxic, but they have lost this response. And I'll show you just briefly in the other slide, in the next slide. But I wanted to touch on, in this case, there's not a hyper-responsiveness that we see. So again, just to give you an example of what we were doing, in this case we have the control and IUGR fetuses, and they're either intact or have an adrenal demodulation. And you can see if we look at the intact IUGR, we have high catecholamine concentrations, and the adrenal demodulated animals have low catecholamine concentrations, and their insulin secretion response is increased to that, similar to what we see in the control adrenergic animals. Although there seems to be some positive aspect to having an intact adrenal gland if you compare the controls to the control demodulated animals. So it seems like if these fetuses have high catecholamines for a long period of time, when that catecholamine response is inhibited or removed, they develop a hyper-responsiveness. We've gone and done early neonatal studies. So we've taken lambs, we've now decreased the heat stress slightly, but we still see a 35% reduction in birth weight, and looked at the insulin secretion response with a square wave hyperglycemic clamp at 8 days of age. And we see a significant increase in that first phase insulin secretion. This puts a challenge to the dogma that intrauterine growth restriction leads to long-term beta cell dysfunction. But this may be just one of the steps in the progression to beta cell dysfunction. And I'll talk about one of the reasons for this in a few minutes. So this led us to the next study of, is this really norepinephrine or is there something else going on? And again, to try to avoid the placental insufficiency and all the complications that go along with that, we decided that we would just infuse norepinephrine, increase the catecholamines, and chronically suppress glucose-stimulated insulin secretion, after which we can remove the infusion or stop the infusion and decide whether the insulin secretion is enhanced. So we would bring in, previously, normal fetuses. I mean, there's nothing wrong with them. We bring them in around 120 days, acclimate them, surgically place the catheters within the venous and arterial circulation, and then let them recover for 4 or 5 days and begin our infusion, and then study the animals either during the infusion or post-infusion. And just to kind of give you an example, we are increasing our norepinephrine concentrations to levels that are very similar to that of what we would see in those IUGR fetuses. We've performed in this series of experiments basically 3 glucose-stimulated insulin secretion challenges, one before, 24 hours after, and 5 days after terminating the norepinephrine infusion. And you can see norepinephrine, after the infusion, norepinephrine levels drop, although they don't drop to normal levels immediately. The other aspect of this is, of course, if you inhibit insulin secretion, glucose concentrations increase. So we infused insulin into the mother, which does not cross the placenta, to limit or help maintain euglycemia in these fetuses. So this is just to kind of show you the treatment that's going on during that norepinephrine infusion. Now, I don't want to get bogged down in all the details because we have published this, but if we perform our square wave hyperglycemic challenge or a follow-on arginine bolus during that hyperglycemic challenge to look at glucose stimulated as well as arginine stimulated insulin secretion, what we see is in the norepinephrine infused fetuses, 5 days after stopping that infusion, a hyper-responsiveness of about 2-fold compared to the controls at the same time. So this may actually really be demonstrating that we do have a programming effect, and whether this programming effect lasts long-term is now becoming a question because we're limited to 5 days. We actually know in the lambs by 30 days it begins to fail, that the islets are already failing to secrete insulin in response to glucose. So it seems to be a very transient response, and it might be one that's very clinically relevant because fetal stress or the outcome of fetal stress leads to hypoglycemia early on. So those fetuses will actually be hyperinsulinemic, become hypoglycemic, and a real challenge in trying to stabilize them immediately after birth. One last point on the norepinephrine infused animals. Of course, these islets, everything that I've shown you so far has been infused, has been infusion in vivo strategies. We've actually developed procedures to isolate these islets, look at them in culture, and in the course where we're challenging these islets with glucose, we see a significant increase in norepinephrine in that norepinephrine infused group, now 6 days post infusion. And if we do we look at the inhibitory concentrations, they actually have reduced responsiveness to norepinephrine, but remain responsive at higher concentrations. This is happening not in response to increased insulin, but in response to mechanisms that actually increase glucose or increase their responsiveness or ability to secrete glucose. So we heard a little bit about RNAseq earlier today, and this is now taking those islets from either that placental insufficiency group or that norepinephrine infused group, looking at the transcriptome, identifying those differentially expressed genes, and then comparing those lists across each other. We're able to identify around 1,000 genes in the IUGR, around 300 genes in the norepinephrine infused that are differentially expressed. Approximately 89 of those genes overlap, again beginning to represent norepinephrine-specific genes that are responding in both the IUGR and norepinephrine-infused fetuses. If we take one other layer off and say they have to be going in the same direction, we're now down to 28 genes that may be coordinated by norepinephrine in this chronic situation. I would lose you all in a minute to after lunch naps if I started going through those lists. But what I do want to point to is one specific gene, and that is an uncoupling protein 2. And just to remind you, we have glucose metabolism going on. As it's metabolized to pyruvate, it enters the TCA cycle, goes through the electron transport chain, where then ATP synthase will generate ATP as long as that proton motive force is, positive or creating this displacement. UCP2 will actually short circuit that force, allowing protons to cross and not go through the ATP synthase. So essentially, decreasing UCP2 in both of these models may enhance insulin secretion, but may also increase their challenge to oxidative stress. How am I doing?
I think you have 3 or 4 minutes.
Okay. So the last point, and this kind of sets up what we've done before, is placental insufficiency, that hypercatecholamia leading to development that then suppresses insulin secretion in utero and enhances it postnatally. That begins to beg the question of how do we treat this, or how do we begin to alleviate this stress? And again, going back to our model of the adrenergic demodulation, what I want to point to is we've done these studies under normal oxygen tensions where these IUGR fetuses have decreased partial pressure of oxygen. Whereas if we actually put in a maternal trachea catheter and bleed in oxygen, we can increase her concentrations and also increase the fetal oxygen tension to levels that are similar to or normal, and enhance insulin secretion independent of whether they have high norepinephrine or not. So I think this is exciting. Again, this is very acute. What we want to do is look at more chronic levels to begin to identify mechanisms or interventions that could improve outcomes even for a few weeks in these IUGR fetuses. The last point that I was going to touch on is future areas, and I think obviously understanding neonatal nutrition during preterm birth is critical. Of course, intrauterine growth restriction, if we begin to identify biomarkers, we may get to etiology of the disease, as well as how do we develop interventions. The sheep is really leading the way in some of these aspects of being able to look at the fetal metabolism in utero and then translate it to humans. Gestational diabetes, of course, is going to be important as we look at the number of mothers that are becoming diabetic, and so we need to develop relevant models there. And of course, Medina is one of her factors is going to be endocrine disruptors and how to actually look at their effects in fetal placental action. So with that, I will thank the funding agencies and my lab crew right now and ask for questions if you have time.
Thanks, John. Any questions?
Yep, Tom.
So Sean, I enjoyed your talk. I was involved in a series of studies when I was at Texas A&M with kind of an undernutrition model. And one thing I was always kind of amazed is that if we did enough animals, we would see basically fetuses that responded appropriately, you might say, in terms of a reduction in growth rate. But there were some that were resistant to the undernutrition, even though their mothers were, they were clearly losing a lot of weight.
Right.
So do you see the same thing in yours where some of your fetuses just are resistant to the hyperthermic effects on the mother?
So it's not the fetus. I think it's the ability of heat abatement by the ewe. So Tim Renault has actually published on this where he looked at the severity of intrauterine growth restriction and those mothers that responded immediately to the heat. And when I talk about responding immediately, their temperatures rise about a half a degree and go up and plateau around 39.7, normally being right at 38.9 to 39.1. When they increase rapidly within the first week, those would be the severe or more severe. Whereas if that mother was able to abate that and not have that temperature increase, it seemed like the fetuses were less growth restricted. That's just an example. I think we've found some that obviously are, we find a wide range of those that are 50% versus those that are, 30%. But at the end of the day, with the heat stress model, they always are impacted. Even if the growth isn't impacted, like some of those lambs you saw, they fell into that very normal range. They were similar to the growth restricted animals more so than the control group. And it was clear that they were impacted even though they were able to get to 4 kilos by birth.
Thank you.
So Sean, kind of
Yes.
easier question. Why do you think hypothermia per se results in intrauterine growth restriction?
So the hypothermia is being caused really when the placenta is developing. So the question is whether it's maternal stress, redistribution of maternal blood flow in that early period. You know, Russ Anthony has shown extensively that it reorganizes the vasculature. So it's probably doing something, you know, there's probably a component to that early stress and redistribution of blood flow in the mom in response to the heat stress. But in cattle, that happens, you know, heat stress can affect them even into late gestation.
In cattle, if you put catheters in like you did, they'll usually abort.
Oh yeah, I wasn't going to put catheters in, but I'm saying heat stress leads to fetal growth restriction.
Thanks, our speaker.